Hi, my name's John Downing. I'm a limnologist and aquatic ecologist, and this is a series of lessons or sessions or modules in limnology and aquatic ecology, and this session is on secondary production. Now, you probably already know something about secondary production. Before we talk about that, let me just say that I think that that we've been talking about animals and animal growth and animal biomass and various aspects of the benthos and the zooplankton. And it seemed to me reasonable that we should also talk about secondary production, which is essentially the rate of growth of animal material. Now, there might be a couple of things that you already know almost subliminally about secondary production. You know that primary production is the growth of plants, right? So you might guess that secondary production is the growth of animals because it's the kind of the next step. In a way, it's a little bit counterintuitive because you've probably learned the concepts of, consumers and then, sort of secondary producers and tertiary producers. But in general, animal production is sort of lumped into what we call secondary production because it's not primary, that it's production that's somewhere down the line. Now, if you eat meat or grow meat, or if you've been involved in animal husbandry or something like that, then you rely on growing animals by feeding them plants or something else, and then they grow, and then you use them as food. That biomass that's built, that animal biomass that's built via the consumption of other kinds of material, that's secondary production. And you also know a couple of things. You know that small animals grow into big ones, generally, and this happens for all kinds of things. As animals grow, some of them die. So you've probably studied some aspect of survival curves and population dynamics, but generally, as animals grow into big ones, there's some mortality, and the sort of the sum and the and sort of the integral of that of the relationship between numbers of organisms and their biomass as they change over time gives us an estimate of secondary production. Pretty straightforward as a concept, a little more complicated to actually do. My objectives in this session on secondary production are to learn the concept of secondary production. I think we've already made a little progress on that. And then I'd like you to learn how secondary production is measured, just sort of the basics of it, and then understand the variables that influence rates of secondary production in aquatic ecosystems. This is basically an overall diagram. This is a diagram built by Frank Rigler in the IBP Handbook number 17, and it was also republished in a subsequent version of that. The sort of the brutal way of thinking about what animals do is to think that they actually only do kind of three important things through their lives, although some of us have hopes and dreams and so on. But basically, a cohort of animal does three important things over their lifespan, and that is to grow, to die, and to reproduce. Probably not in that order. Probably grow, reproduce, and then die. Now, how fast those things are done determines the rate of food supply to other trophic levels, and that is why secondary production is important. The definition of secondary production is the amount of animal tissue elaborated per unit time, regardless of its fate. So, regardless of what happens to it, whether it goes to death and detrital material, whether it's eaten by something, where it goes into reproduction, that elaboration of animal tissue is in fact the secondary production. So you can understand a little bit more about how secondary production works by looking at this diagram. Over here we have the food that comes into an animal. This is the ingestion rate that we talked about quite a bit back in the session on zooplankton. So that's the ingestion rate. Food comes in and is used. Now, it's used in a couple of ways. First off, some of it is assimilated here, and some of it's egested. Okay, so this egestion would be sort of fecal material production that you probably are pretty familiar with. So when food gets eaten, it either is assimilated or egested. There aren't too many other choices. So clearly, you could calculate the rate of assimilation as ingestion less egestion, and that would give you the amount that was assimilated. Now something happens with that assimilated material, as it goes into an animal, is that some of it is used for overall maintenance. So some amount of that energy or food material that's taken in is a sort of a loss, really, to overall secondary production, in that it's used for respiration. So this is the amount that it takes to keep that animal tissue alive. And you've probably heard of things like basal metabolic rate and a variety of things like that are the rate of maintenance respiration. And it can be quite a significant quantity. Now, if you take the assimilation and take away respiration from the assimilation, that gives you the rate of secondary production. Now, in effect, we don't do it like this. We don't go around collecting feces and measuring respiration rates to measure the secondary production. There are better ways to do it. But the secondary production is all of that animal tissue that's elaborated, and that animal tissue goes into a variety of things. It goes into individual growth, and it goes into the production of new young. So it's used as growth and reproduction. So some percent of that production goes into each of those. Now, of that production, some of it's lost to a predation rate, some of it's lost to a natural death rate, and some of it is also lost to overwintering eggs. But overall, regardless of its fate, whether it goes into growth, production of new young, to loss to predators, loss to detrital microconsumers as sort of natural death rate, or to overwintering eggs, this whole quantity is the secondary production and is potentially usable by a subsequent trophic level. So looked at as sort of an overall equation, the production rate is ingestion less egestion less respiration will give us secondary production. Now, I said before, we don't measure it like that. It's way too hard to measure all of those sorts of things. And all we're really wanting to know something about is the overall aggregate rate of growth of animal tissue by, say, a cohort of animals. Now, cohort, you've heard people talk about your cohorts, right? People who belong to your same age group, basically. And so when we think about a cohort of animals, we're thinking about animals that are all sort of born at the same time and go through life. It's sort of in synchrony. So that's the cohort concept. So just looked at in a different way, this is secondary production in an eggshell, basically. We have two important processes here, really, in measuring secondary production. One of them is animal growth, and one of them is mortality. Growth is obvious. We go from an egg, which has pretty small dry mass, to a hatchling. A hatchling grows and pecks around and eats a whole bunch of different things. And these are obviously chickens, not aquatic animals. I mean, but it's the same is true for aquatic animals. So the chicks grow, and they eventually grow into adults. So that's sort of they go through various life stages of growth. And looking at copepods and other kinds of aquatic organisms, we also think about life stages. So if you get more deeply into this, you'll see that parallel. The other thing that's going on as the organisms are growing is they're dying. Not all of the eggs hatch, not all the hatchlings live, not all chicks become adults, and eventually even adults stop growing and eventually die. So two processes going on, and they are growth and they are mortality. Now, for a cohort of organisms, this is kind of the simple way to measure it. If we have a bunch of organisms who are all, say, hatched at a given time, let's say, at a very low body mass, and we have 10 of them, let's say, that are born or hatched at that particular time. What happens is over time, as sort of we pass down this line of time, this would be actually a timeline here. So you, in a way, have three dimensions. We have individual mass, we have body size, numbers. This is body size, this is numbers, and here we have some sort of mortality rate over time. So what happens is we lose some of those organisms. Here we have the death of one, death of another, death of another. We see this ratcheting downward here. And eventually, all of those animals from that cohort will be dead. As they are dying, they are all growing. And so if we're interested in the secondary production rate between this time, which is the time that those organisms are born, and this time here, which is some time in the future, let's say when they have a body mass of 5 units, whatever that might be, the secondary production is essentially the integral under this curve, and that gives us a number times grams per unit or per organism, and then that will give us overall mass of production. Very simple kind of calculation. Now, the reason we have stair steps shown on here and some big dots is that this is kind of an attempt to show how we might be able, if we had all the data in the world, to follow every organism in a population as it dies, and then we could measure this integral underneath this curve. In effect, what we do is we go out and sample at various times. We say, okay, the number we have there is this, their average mass is this, the number we have is this at a different time, and the mass is this, and so on. And then use that integral to calculate secondary production. A fairly simple conceptual calculation. Now, there are lots of other ways. I should go back here. There are other things we have to do in order to measure this, because we can't always find a given cohort and follow it through its life. Sometimes we do this with various shortcut methods that are a little bit more advanced that we don't really need to treat at this time. Now, a lot of factors influence secondary production, and you can guess what they might be simply based on your understanding of physiology of mostly poikilothermic animals. In fresh inland waters, we have very few homeotherms, although there are some in some systems. But so most of these are poikilothermic, so they respond to temperature, for example. So here's a graph showing for a bunch of different kinds of organisms the temperature and the turnover ratio. Turnover ratio is essentially the ratio of secondary production to standing biomass. So you can kind of see this turnover ratio as a percentage yield on an investment of biomass. That's how much it's giving back per unit time. And this may be a turnover ratio for a year or a month or a day. In this case, I think it's annual. And so what we're seeing is turnover ratios or percentage yields in the neighborhood of 2, 5, 10, 15 times, 15-fold the return on biomass. So I think you can tell that these things are growing pretty rapidly. But as temperature gets lower, the turnover ratios in secondary production tend to decline so that you don't have very high rates because animals are not growing very fast. Another factor that influences secondary production is primary production and sort of a detrital rain of material, the supply of new energy coming to these organisms. What you see in this graph is for a bunch of different zones in various areas, lakes in various places like the Arctic, temperate zone, and so on. Here we have the production of macrobenthos, and here it's measured in kilojoules per square meter per year. So we said before when we talked about primary production that you can measure production in units of carbon and units of energy and so on. In this case, it's units of energy. And this is the primary production rate measured in the same sorts of units. A couple of things to take away from this graph. One is, as there's more primary production, clearly there's a whole lot more macrobenthos production being fueled by that detrital rain of primary production. Another thing to look at is to cite up these primary production rates. And so we have 100 units of primary production, and somewhere in here we're finding about 10 units of macrobenthos production. So about a 10% loss 90% loss of production, sort of from one trophic level to another. Let's take another one. If we say 10,000 units of primary production, we get over here to about 100. So we've got 100 out of 10,000. So a fairly substantial loss rate of that primary production, especially where primary production is very high. Now, a high standing biomass usually gives us high production. We see the same thing assumed for primary production, right? Because we always say primary production increases with chlorophyll A concentrations in phytoplankton, and those chlorophyll A concentrations are a surrogate measurement of biomass. Same is true, really, for annual production of various kinds of invertebrate organisms in lake ecosystems. This is the mean annual biomass, or annual mean biomass, actually, and this is the annual production. And you can see that as we have more biomass in a given place in a lake ecosystem, we tend to get much more production. On the other hand, the exponent here, or it's not an exponent, this would be a slope in a logarithmic relationship, which turns into an exponent. It's lower than 1, which implies that as mean annual biomass, or annual mean biomass, gets very high, we get sort of less secondary production back per unit. Now, this may be due to things like competition or maybe interference, but generally production is a little bit less efficient at higher biomass. We can put all these factors sort of into a couple of graphs, and these are three-dimensional graphs. So I'll spend a little bit of time on them, trying to show you how to read them. But these are something like the turnover ratio that I talked about a minute ago. This is actually production to biomass ratios. So the ratio of production to the standing biomass. And so it's the proportional yield. And again, you can see this as essentially an interest rate. And if the P to B ratio is 1, it means you're yielding every year an amount of secondary production that's equal to the biomass. A number of 2 means you're doubling, the production is double the biomass, and so on and so forth. So the bigger the number, the higher the proportional yield from that particular population. And this is put together from a lot of data from a lot of ecosystems. So down here we have the body mass of organisms. That's their body size. These are really big things over here, really little things over here. And this is the standing biomass. And we already know from the previous graph that we should expect that as we increase the standing biomass, we will get a somewhat lower proportional yield because of that, whatever it was, interference or competition that's going on. And in fact, that's true. And it drops pretty rapidly. And these are in grams, in logarithms of grams. So this is 1 gram, and this is 100 grams, and this is 100th of a gram. And so you can get a sense of how much. This is a range of variation of 6-7 orders of magnitude, and the production to biomass ratio, or the proportional yield, essentially for a small organism goes from very high, 100 times its biomass produced every year, to maybe 8 as you get to a very high biomass. So there's a very strong tendency to decelerate production at very high biomass. Likewise, if you look at the body size, the bigger things tend, as I've said before in previous sessions, big things do things more slowly, little things do things more rapidly. So clearly, the very large organisms, the largest organisms in this set of populations, or communities actually, tend to have very low proportional yields for a given overall biomass. The other factor that we need to integrate into here is temperature, and you can see how temperature changes secondary production for a variety of different sizes of organisms. But in general, as one increases temperature from very low temperature here, let's take this size of an organism. From very low temperature, we're at a turnover ratio or proportional yield of about 2 times the material produced per year as the average standing stock. And we move over to higher temperatures up in 24 range, we're up somewhere around 25 times that biomass produced. So you can see why tropical systems are highly productive, or we get this very large increase in growth of new food for upper trophic levels when the temperature gets warm. Now the benthos, so benthos and zooplankton production can be quite substantial, and they can be a very large fraction of the net carbon inputs, and carbon here again is a surrogate of productivity. Here we have the production of macrozooplankton and macrozoobenthos as a percentage of net carbon input into this production. You can see that these numbers are fairly high. They can be as much, almost a quarter of that carbon input can go into either macrozooplankton or macrozoobenthos. Likewise, zoobenthos can be very important. It's less easy to study, but this is the percentage. This is the percentage of production that's represented by the zoobenthos, and you can see that it can be a very large number depending on the lake ecosystem that you are considering. So the summary on secondary production, a fairly short session, is that secondary production is the rate of biomass growth of animals regardless of its fate. Secondary production is measured by following population dynamics of things like zooplankton, benthos, and fish, and the production varies with a variety of different, very logical kinds of variables, including things like food availability, body size, and standing biomass, and temperature.